For decades, scientists knew that something was hiding inside Tibetan DNA. It was not a disease, not a random mutation, and not simply the result of life at high altitude. Every time researchers tried to rebuild the ancestry of the people living on the Tibetan Plateau, most of the genetic pieces fell into place. A large part pointed toward ancient populations from northern East Asia. Another small but important part carried Denisovan inheritance, including the famous high-altitude adaptation connected to the EPAS1 gene. But one piece refused to fit. It came from a population no one had clearly found. The signal appeared again and again, like a shadow trapped inside living genomes. Scientists could measure it, model it, and estimate that it was ancient, but they had no confirmed cemetery, no known culture, and no skeleton they could point to and say, these were the people. In genetics, a population like this is called a ghost population. It does not mean spirits or anything supernatural. It means the population is visible only through the ancestry it left inside others. And for years, one of the ghosts inside Tibetan ancestry had no face. Then researchers examined the remains of a woman buried in Yunnan, in southwest China. Her grave was not royal. No gold crown announced her importance. No weapon suggested that she had been powerful. She had been placed beneath later burials at the Shingi archaeological site, almost as if history had pushed her deeper into the ground and forgotten her. Yet inside her bones was something more valuable than treasure. Her genome carried a deeply separated ancestry that had never before been sampled so clearly in an ancient person. And when scientists compared it with modern DNA, part of the pattern led toward Tibet. The woman is known by the scientific label Shingi An. She lived roughly 7,100 years ago, during the early Neolithic, before cities, kingdoms, or written records appeared in that part of the world. Her DNA became the most mysterious result in a 9-year project that recovered genome-wide information from 127 ancient people across 17 sites in Yunnan. Those individuals range from about 7,100 to 1,400 years old. Together, they revealed that southwest China was not an isolated edge of prehistoric Asia. It was one of its most important crossroads, where deeply different populations met, mixed, moved, and sometimes vanished. But Shingi An did not fit neatly among the other ancient people. Her ancestry was so deeply separated from most present-day East Asians that researchers gave it a new name, Basal Asian Shingi ancestry. The word basal can sound dangerous if misunderstood. It does not mean primitive, less intelligent, or less human. It means that the lineage branched away early from the other Asian lineages being compared. Genetic modeling suggested that the population represented by this woman had separated from other Asian populations at least 40,000 years ago. Somehow, part of that lineage survived in southwest China until the Holocene, long after the last ice age had begun to fade. That discovery immediately created a dramatic question. Had scientists found another human species? Was this woman part Neanderthal, part Denisovan, or connected to some unknown archaic human that modern Tibetans had interbred with? The answer, based on the evidence, is no. The study did not identify a new species. Shingi An was a modern human. Her ancestry was deeply diverged within the modern human branch, not proven to come from a separate species. The mystery is not that she was non-human. The mystery is that an ancient modern human population survived for tens of thousands of years, disappeared as a distinct community, and left part of its ancestry inside people living on the Tibetan Plateau today. This matters because Tibetans really do carry DNA inherited from Denisovan-related humans. The most famous example is the EPAS1 haplotype, which is associated with the body's response to low oxygen. Research has shown that this unusual genetic version entered modern human ancestors through interbreeding with Denisovans or a closely related population. It later rose to high frequency among Tibetans because it helped people survive at extreme altitude. But the Denisovan EPAS1 story and the Basal Asian Shingi story are separate. One involves archaic introgression. The other involves a deeply separated population of modern humans. Combining them into one mystery species story would be exciting, but wrong. For years, geneticists trying to explain Tibetan ancestry had faced a stubborn problem. Most of the ancestry in ancient and modern plateau populations could be connected to people related to ancient northern East Asian populations, including Neolithic communities associated with the Yellow River region. One major genomic study estimated that ancestry related to northern East Asian farmers made up around 84% of the gene pool in some Tibetan groups. Yet another component remained more difficult to identify. Official summaries of the new Yunnan research describe roughly 80% of plateau ancestry as connected to northern populations, dating between about 9,500 and 4,000 years ago, with the remaining portion linked to a previously unclear ghost source. Shingi An did not perfectly solve every part of that missing 20%, but she provided the first clear ancient representative connected to one of those ghost sources. Her genome showed affinities with the deeply diverged ancestry found in ancient plateau populations and preserved in modern Tibetans. This was not a direct claim that Tibetans descend mostly from one woman or from one small group in Yunnan. It meant that a population related to hers contributed ancestry to populations involved in the formation of Tibetan highlanders. The ghost had not been fully resurrected, but for the first time, science could place part of it in a real body from a real archaeological site. The story became even older when researchers compared Shingi with another ancient individual known as Longlin. Longlin lived in Guangxi around 11,000 years ago and had already puzzled geneticists because of the unusual ancestry inside the genome. The new analysis showed that Longlin carried a mixture that included ancestry related to the Shingi lineage and another ancient southern component. This meant the deep lineage was not limited to one woman at one site. It had existed across a wider region of southern China before or during the early Holocene. But then something happened. In later Guangxi populations, dating to roughly 8,000 to 6,000 years ago, the Xingyi-related signal fell sharply. By historical times in the sampled groups, it had disappeared. And yet, far to the west and north, a related signal survived in populations connected to the Tibetan Plateau. That creates one of the most important mysteries in the entire study. Why did this ancestry fade in much of southern China, but remain inside Tibetan highlanders? Did people carrying it move toward the plateau? Did the DNA travel through a chain of mixtures between neighboring communities? Did the lineage survive in isolated mountain valleys that have not yet been sampled? Or did a sister population related to Xingyi but living closer to Tibet become the real source? The genetics shows a relationship. It does not yet provide a complete route. The geography of Yunnan makes several possibilities believable. The region is filled with steep mountains, narrow basins, high plateaus, and deep river valleys. Great rivers such as the Upper Yangtze, Mekong, Salween, and Red River connect southwest China with Tibet, Sichuan, Southeast Asia, and the coast. A mountain ridge can isolate one community for generations, while a river corridor can guide movement across enormous distances. This landscape can preserve old genetic differences and then suddenly bring populations together when climate, trade, farming, or migration changes. Yunnan was both a refuge and a gateway, a place where ancient lineages could hide and a place where they could spread. Imagine the world of Xingyi. The climate, vegetation, and animal life were different from today. Farming had not yet transformed the area completely. Isotope evidence connected to the Shingi sequence suggests that the earliest inhabitants relied mainly on hunting and gathering. They would have understood seasonal plants, animal movements, water sources, and mountain routes that leave almost no written trace. The woman may have belonged to a small mobile group or to a community that returned to the same place at different times of the year. Her bones preserve the genetic history of her population, but almost nothing about her personal life. We do not know her name, language, clothing, cause of death, or what she believed. The absence of grave goods makes her discovery more haunting. Archaeologists often tell history through objects: pottery, tools, jewelry, houses, weapons, or monuments. Xingyi was not surrounded by anything that announced a special status. If her DNA had not survived, she might have remained one more silent burial beneath later graves. Instead, the least visually impressive grave became the key to a lineage that had separated at least 40,000 years ago. The discovery is a reminder that the most important archaeological object is sometimes not an object at all. It can be a few damaged molecules inside an ordinary bone. Ancient DNA from southern China is extremely difficult to recover. Warmth, moisture, acidic soil, bacteria, and thousands of years of chemical change break DNA into tiny fragments. A bone can look solid, while the genetic information inside it has almost completely disappeared. Researchers have to work in clean laboratories, sample dense skeletal material, identify characteristic damage patterns, and estimate modern contamination. Even after a successful extraction, the genome may be incomplete. That is why the Yunnan project took years and why 127 ancient genomes from 17 sites represented such a major advance for East and Southeast Asian prehistory. One unusual genome can always raise doubts. Perhaps the person was a recent migrant. Perhaps contamination affected the result. Perhaps the statistical model lacked the right comparison. The strength of the Yunnan study came from its scale and chronology. The researchers did not examine one skeleton in isolation. They compared individuals across nearly 6,000 years and multiple regions of Yunnan. They placed them alongside ancient genomes from northern China, coastal southern China, Guangxi, Southeast Asia, the Tibetan Plateau, and other parts of East Asia. Xingyi remained different even within that large framework. The lineage was not created by an empty database. It stood apart after extensive comparison. Still, one major limit must remain clear. Xingyi is only one directly sampled individual, carrying a very strong form of this ancestry. When scientists say she represents a ghost ancestor, They mean she provides the closest sampled genetic connection so far, not that every missing ancestor has now been identified. The study also revealed that Yunnan did not contain just one deep lineage. Ancient people from central Yunnan between about 5100 and 1400 years ago carried another distinctive ancestry, named Central Yunnan ancestry. This lineage was different from both northern and southern East Asian ancestry, and appears closely related to ancestry found among many present-day Austroasiatic-speaking populations. Austroasiatic languages include Vietnamese, Khmer, and languages spoken by the Wa, Blang, and numerous communities across Southeast Asia, southern China, and northeastern India. The new genome suggested that central Yunnan may have been one important early region in the history of those populations. That second discovery strengthens the larger point. Prehistoric Yunnan was not occupied by one stable people waiting to become a modern ethnic group. It contained deeply separated lineages and later mixtures. Western Yunnan received strong ancestry linked to northern East Asian populations and communities related to Yellow River farmers. Eastern Yunnan showed mixtures involving southern East Asian and central Yunnan ancestry. Central Yunnan preserved a lineage with connections to later Austroasiatic-speaking populations. Beneath all of these later patterns, Shingi revealed an even deeper layer. Each region of Yunnan carried a different history, and those histories changed repeatedly through time. The archaeological sequence at Shingi offers another surprise. Isotope and subsistence evidence suggest a long transition from hunting and gathering toward farming. Culture moved, but the population remained. The same families may have watched hunting grounds become fields over many generations, adapting to a new economy without disappearing. Yet Shingi lived before that stable farming era population developed. Genetic change occurred between the earliest hunter-gatherer layers and later inhabitants. That means her deeply diverged ancestry did not remain unchanged at the site forever. New ancestry entered, older ancestry mixed, and the population structure shifted. The lineage represented by the woman survived in reduced or transformed form, rather than continuing as a pure, isolated group. This is how ancient populations usually disappear, not always through massacre or sudden extinction, but through mixture, demographic expansion, and the gradual loss of a distinct genetic profile. The word pure has almost no useful place in this story. The population represented by Shingi was deeply separated, but it was not frozen outside human history. Longlin already showed that Shingi-related ancestry mixed with another southern lineage by 11,000 years ago. Later, plateau populations mixed it with northern East Asian ancestry. Modern Tibetans carry multiple ancestral sources, including different levels of lowland and highland-related ancestry. Every stage involved contact. Deep ancestry does not mean purity. It means that a branch remained distinguishable long enough for its signal to be detected. The route toward Tibet may have followed the eastern and southeastern margins of the plateau. Yunnan, Sichuan, Qinghai, and the highlands are connected by river valleys and mountain corridors. People could have moved gradually, one community at a time, without any single dramatic migration. A marriage between neighboring groups can carry ancestry across a boundary. Repeated over centuries, those small movements can transport genetic signals hundreds of kilometers. By the time descendants reached high-altitude regions, the original community name, language, and identity could have disappeared, while part of its DNA remained. Earlier ancient genome studies show that the formation of Tibetan populations involved strong ancestry connected to northern East Asia. Yellow River-related ancestry moved westward and upward, likely alongside expanding communities and technologies. But genetic evidence also points to older, deeply separated ancestry inside highlanders. The new Shingi genome gives researchers a better source for part of that deep layer. It suggests that Tibetan formation was not simply a northern farming population climbing into an empty landscape. Northern migrants encountered or absorbed ancestry connected to populations with much older roots in southern or southwestern East Asia. The plateau was never empty. Denisovan-related people occupied Baishia Karst Cave on the northeastern Tibetan Plateau long before the appearance of modern Tibetan populations. The Xiahe mandible, identified through ancient proteins, showed that Denisovan-related humans lived at high altitude around 160,000 years ago. Sediment DNA and later fossil evidence indicate repeated Denisovan presence over a long period. These archaic humans hunted or processed animals in an environment that would challenge most people today. Their history shows that high-altitude Asia was part of the human world far earlier than once believed. Modern humans also reached the plateau early, though archaeologists still debate the permanence of those occupations. Stone tools from Nuea Devu indicate human presence in the plateau interior tens of thousands of years ago. Later communities developed increasingly stable ways of living at altitude. A recently studied site at Mabuko, around 4,146 meters above sea level, provides evidence for a sedentary indigenous population between about 4,400 and 4,000 years ago. Those people mainly carried southern plateau ancestry. showing that highlanders had established lasting communities deep inside the plateau before the historical era. Survival at such altitude requires extraordinary adaptation. Other genes, developmental responses, culture, diet, clothing, architecture, fuel, herding, and knowledge all mattered. DNA did not conquer the plateau alone. Human communities survived because biology and culture worked together. The Denisovan-derived EPAS1 haplotype is one of the most dramatic examples of useful archaic DNA in living humans. Denisovans were not ancestors of all Tibetan DNA. They contributed a small segment that became highly valuable under high-altitude selection. The Basal Asian Shingi lineage contributed a different layer of ancestry. It may have shaped the broader population history, but the current study does not connect it directly to EPS1 or prove it provided a high-altitude advantage. These tools are powerful, but they are not perfect. A model can only use the populations available in the database. If the true source has never been sampled, researchers use the closest known relative. Shingi Ian may therefore be a representative of the ghost ancestry, while the exact source population remains buried somewhere else. That uncertainty makes the discovery stronger, not weaker, because it creates a clear path for future research. Scientists now know what kind of ancestry to search for. Older skeletons from Yunnan, Guangxi, Sichuan, and the plateau margins may reveal whether the lineage was widespread. Genomes from between 7,000 and 4,000 years ago could show when Shingi-related ancestry mixed with northern East Asians. Samples from eastern Tibet might identify the corridor through which it entered plateau populations. A single tooth from the right place and period could turn the current connection into a detailed migration map. For the viewer, the phrase ghost lineage may sound like one hidden tribe waiting to be discovered. In reality, the ghost may have consisted of many related populations spread across different valleys. They may have spoken different languages and lived in different ways while sharing deep ancestry. Some branches may have moved toward Tibet, others may have mixed into southern populations and disappeared. Shingi Arinyam could be one member of a complex network rather. This is why scientists usually say ancestry related to Shingi Ayen, not ancestry directly from Shingi Een. She was one person. Modern populations do not descend genetically from every ancient individual who shares their ancestry. She may have had no surviving children at all. What matters is that she belonged to a population genetically close to a source that did contribute. Her genome acts as a signpost pointing toward a larger community. The discovery also changes the meaning of extinction. The Shingi population did not need to be destroyed completely for the lineage to disappear. If a small group mixed into a larger population, each generation would carry a smaller average share of the old ancestry. Random inheritance could remove it from some descendants while preserving it in others. After thousands of years, the population name and culture might vanish, while scattered DNA segments survived. In Guangxi, the signal became rare and disappeared from later sampled groups. On the plateau, it was preserved more strongly because of different population histories. The discovery also warns against treating ancestry as identity. A modern Tibetan person is not 20% ghost population in a cultural sense. Genetics does not divide a living person into separate ancient tribes. Every genome is a mosaic, but identity comes from language, family, community, history, and personal experience, as well as ancestry. The Shingi-related component is one layer of biological history. It does not define who Tibetans are, nor does the deep split make the Shingi lineage more ancient in a way that places it above others. All living humans trace their ancestry through populations that existed tens of thousands of years ago. A lineage can branch early without being unchanged or more primitive. Every population alive today has evolved for exactly the same amount of time. The Basal Asian label describes a position in a genetic tree, not a stage in human progress. The most powerful part of the story is the contrast between the scale of the history and the silence of the burial. The lineage may have survived for more than 30,000 years after separating from other Asian populations. It endured climate swings, the end of the Ice Age, new migrations, and the spread of farming. Yet the woman who revealed it had no inscription. No one recorded the name of her people. The entire discovery depended on molecules so small they could not be seen when the grave was opened. For thousands of years, her skeleton remained beneath later burials while the world above changed. Hunting communities became farmers. Villages grew. Languages spread. Populations moved through Yunnan. Farther west, highland societies developed ways to survive on the Tibetan Plateau. Empires rose and disappeared. The ancestry related to her people continued moving silently through human bodies, broken into smaller pieces by recombination, until modern genetics finally recognized the pattern. The result is not a final answer. It is the first strong chapter in a larger investigation. Researchers still need to discover the population that directly formed the deep component in early Tibetan genomes. They still need to date the mixture with northern East Asian-related groups more precisely. They need to know whether the ancestry moved north once or arrived through several waves. They need to test whether later population movements carried Shingi-related ancestry back into southern Tibet, as more recent genomic studies have suggested, for periods after 4,000 years ago. Every new ancient genome could change the route. A sample from eastern Sichuan might show an intermediate population. A burial from the plateau margin might reveal an early mixture. An older Yunnan skeleton could push the lineage deeper into the Ice Age. A cave in Guangxi might show why the ancestry declined there. This is what makes ancient DNA research so suspenseful. A model that looks convincing today can become only one branch of a more complex story tomorrow. Still, several conclusions are now well supported. Their descendants survived in the complex mountain landscapes of southern China. Some mixed with other groups and faded from the record. Others contributed ancestry to populations involved in the formation of Tibetan highlanders. Did they ever climb onto the plateau themselves? The current evidence cannot say. Their DNA may have traveled through people who never saw the highest mountains. A lineage can move through marriage and gradual migration without the original population relocating as one group. Or perhaps future skeletons will reveal Shingi-related communities, living much closer to Tibet than anyone now knows. That is the question waiting beneath the next excavation. That may be the most unsettling lesson. The people we know from archaeology are only the ones whose remains survived, were found, and still contained readable DNA. Entire populations could have lived for thousands of years in warm regions where genetic material decayed completely. Their only surviving evidence may be a faint statistical signal inside living people. Shingi was found. Many others may never be. For decades, scientists could see the shadow inside Tibetan DNA, but could not identify the people behind it. Now the shadow has moved closer to the light. It leads away from the plateau, through mountain corridors, toward a quiet burial in Yunnan. Populations met. Ancestry mixed. People moved. Languages changed. The old lineage lost its independent name and became one component inside a new population. No written record captured the moment. Only DNA remembers that it happened. And that leaves one final question. If a single woman buried without treasure could reveal a human lineage hidden for tens of thousands of years, how many other ghost populations are still inside us right now? How many people disappeared from history without disappearing completely from the human genome? The answer may already be present in living DNA, waiting for one ancient skeleton to make the invisible real. Subscribe to past proof because the past is gone, but the evidence remains.